Hello, everybody. Welcome to our GlycoNet ACS joint webinar series. It is my profound pleasure to introduce Professor Lisa Willis, who is an assistant professor here at the University of Alberta. Dr. Willis received her bachelor's in science and biochemistry at the University of Victoria and her PhD in molecular and cellular biology in the laboratory of Dr. Chris Whitfield at the University of Guelph. I hope I'm pronouncing that correctly. She did her postdoctoral fellowship with Dr. Mark Nitz in the Department of Chemistry at the University of Toronto. As you can see, she's very much a Canadian with all of the various Canadian universities, hitting really the highlights of this amazing carbohydrate community here, where she received the prestigious Banting Postdoctoral Fellowship. We were lucky enough in 2019 to have her join us here as an assistant professor in the Department of Biological Sciences at the University of Alberta, and she's also an adjunct professor in the Department of Medical Microbiology and Immunology here. And she is going to tell us all about her work, glycoengineering polysialic acid, and the insights into sex differences in human polysialic acid. And I'm very happy to get to listen to her latest research. So take it away, Lisa.
Thank you very much for that introduction. That's not what I want a laser pointer. Okay, I'm really excited to be talking with everyone today about what we're doing, trying to study polysialic acid. I would like to start with a land acknowledgement. The land acknowledgements are really important in Canada. In Canada, we're undergoing a painful but necessary process of reconciliation with the Indigenous peoples who have lived on this land for many thousands of years. And as someone who lives in Canada and has studied in Canada, it's my responsibility to understand the issues and to participate in the process of reconciliation to the best of my ability. Another important component of the land acknowledgement is to actually acknowledge the land itself. And I am committed to respecting and preserving the land for future generations. I am thrilled to be talking to a glycobiology audience. I don't think I have to convince anyone that glycobiology is awesome and fascinating and frustrating and just so much fun. Just to put everyone in the context of what we're going to talk about, I'm very interested in sialic acid, specifically polysialic acid. So we have sialic acid, alpha-2,3 and alpha-2,6 linked sialic acid on N and O linked glycans. That's the context in which I'm going to be talking about sialic acid today. That first sialic acid, if it's not elongated with any alpha-2,8 linked sialic acid, is called monoSia. And then you can get the addition of one alpha-2,8 linkage, so that's a diacylated species, and then multiple up to about 400 potential sialic acids in this long linear chain. And so that gives you oligosialic acid and polySia. The differences between these two are they're based on what an antibody binds to. An antibody binds to a minimum of 8, so that's considered polysialic acid. Whether there are functional differences between oligo and polysia, we don't really know. We don't understand if 50 sialic acids, polysia of a polymerization of 50, it has functional differences from a chain that is 400 long. We really don't know. And in contrast to monoSia, which has been very well studied, and we really understand a lot of what that does and how that functions. Polysialic acid has been less well studied, partly because it's not as common. And I'll show you that on the next slide. There are less than half a dozen proteins in humans for which there's really good evidence that they're polysialylated. And we don't have very good tools to study polysialic acid. And that's really what we are trying to do, is to advance what we know about this really cool sugar by developing new tools. So the glycan is not just found in, but is crucial for proper functioning of the nervous, immune, and reproductive systems in healthy human adults. Its role, I've just put two sort of themes here. One is that it increases cell migration, and the other is that it seems to decrease the immune response. So when you have polysialic acid, for example, cells are less phagocytic. When you remove polysialic acid, you get an increase in the secretion of pro-inflammatory cytokines. But we don't really understand the broad strokes of what polysialic acid does beyond this. It's been a bit of a mystery. And we really know very little about the mechanism by which polysialic acid does any of these things. It used to be thought for the longest time that it was just a physical barrier. Because of its large size, it's highly hydrated, it just formed a physical barrier between molecules like adjacent cell membranes and pushed those membranes apart, and that's what allowed cells to migrate. We also know that it has a masking effect, similar to monoSia. Polysialylated proteins in circulation are longer. They have longer half-lives than those that are not sialylated. And that could be important when we start to think about what polysialic acid does in the context of immune function and some of the diseases in which aberrant polysialation plays a role. But recently we've been, and this is mostly the work of Professors Sato and Shiro in Japan, really done some very cool work suggesting that polysialic acid participates in receptor ligand interactions similar to other sialicides. And in fact, maybe in the context of the immune system, which is where we're really interested in it, may be a kind of immune checkpoint, and that's how it attenuates the immune response. This is really fascinating to me. But again, this is the bulk of what we know. And we really, I think we need to do better to understand more. The glycan is dysregulated in people with cancer, regardless of what tissue the cancer of origin is. Expression of polySia in that tissue corresponds to metastasis, an increase in metastasis and poorer prognosis. And it's also dysregulated in people with mental health disorders. Okay, so the first thing I'm gonna show you is how we figured out how to quantify polysialic acid. Previous to this, it wasn't possible to actually quantify the glycan in any meaningful way. And we can quantify this glycan in a complex mixture with no pre-treatment, no delipidation, no acid treatment, none of the things that you would normally need to do in order to measure polysialic acid concentration. And it's just a simple ELISA. What we've done is to put the Polysialectin. This is the endosialidase protein that normally hydrolyzes polySia, but it's got 2 mutations, which make it a high affinity lectin for polySia. We adhere that to the plate. We put in our polysialylated sample. It binds. We wash away. The beautiful thing about this lectin is that it is resistant to detergent. So your sample can be in detergent, unlike antibodies. It's really robust. And then we can come in with a primary antibody that recognizes polysialic acid, a secondary antibody conjugated to horseradish peroxidase. And if we add TMB, the horseradish peroxidase turns it blue, and we get a really nice signal for human serum, which disappears when we pre-treat with the active endosialidase to cleave polysialic acid. We also see polysialic acid in fetal bovine serum. So we played around with this assay to get it to a point where it was linear, which is fantastic. And then we started to analyze the concentration to determine the concentration of polysialic acid in human serum. And we disaggregated by sex, as everyone should. And we actually found that males have a higher concentration of polysialic acid in their serum than females do. These are just 20-year-old healthy donors, so not representative of the whole population. We're working on that. But it's really interesting that males have a higher concentration of a glycan that turns down the immune response. We know that the immune response in males is less easily activated than in females. And that's why males are more prone to get non-reproductive cancers and a number of infectious diseases. On the other side of things, females have an easily activated immune response and are more likely to have a dysregulated immune response, leading to autoimmune disorders. So this is really interesting, correlation, causation, maybe not causation, but at least participation in this phenomenon, we don't really know. But I find it fascinating. The other cool thing about this ELISA assay that we've developed is that you can also, instead of coming in with an antibody to the glycan, come in with an antibody to the protein component of the glycan. And in this way, help identify and quantify polysialylated proteins. Polysialylated proteins are really hard to detect by Western blotting. Because polysialic acid is so long, you would normally see a mobility shift if you're looking for your protein of interest. The problem is that the glycan has to be long enough to actually make the protein shift. And if your protein is already 150 kilodaltons, it needs to be a lot of polysia to make it shift. Also, if not 100% of your protein is polysialylated, then you won't actually see that shift to the higher molecular weight. You know, if only 5% of your protein is polysialylated, you might not see that, especially since the length of polysialic acid on a given protein can be quite heterogeneous. And so what you see is a smear instead of a nice higher molecular weight tight band. So this is a really convenient way to analyze and indeed quantify, if you have a good standard, polysialylated proteins. So we did this with NCAM. NCAM is the best studied polysialylated protein, also known as CD56. This is the neural cell adhesion molecule, so very important for the function of polysialic acid in neurons, neurite outgrowth, synaptogenesis, all of that kind of thing. And then it is the defining marker of natural killer cells in the human immune system, where it's known as CD56. We took human serum, and again, instead of looking at polysialic acid, we came in with a primary antibody to CD56, and we see really good signal, which disappears when you treat with the polySia hydrolase end-to-end. That's great. So we looked at those same individuals, the concentration, and this is relative concentration because we don't have an absolute standard of polysialylated NCAM in human serum. And while females definitely have a little bit less, it wasn't significant. So it's not clear at this point if the length of the glycan in males is longer, and that's why we see a higher polySia signal, or if there are just additional polysialylated proteins in serum, and that is my suspicion, additional polysialylated proteins in serum, which also have a skewed sex difference in their concentration. So we took this assay and we analyzed a panel of cancer cell lines to see what we could see. And polysialic acid was actually more common than we were expecting. So it's present in all of the immune cells, as you might expect, because it is present normally in the immune system, in all leukocytes, as far as we're aware. Maybe not B cells, maybe just low concentration B cells, we're not sure. But it was also present in MCF7, breast cancer cell line, and to a lower extent, PC3 cells, which are prostate cancer cell line. What's interesting, though, is that with the exception of KG1A, really not a lot of NCAM, suggesting that there are other proteins that are polysialylated in these cell lines. And that brings us to the second part of the second method that we developed. And it's a way to identify polysialylated proteins. So it's a new method. It's a metabolic labeling method. We took NK92 cells, so this is a natural killer cell line. It should have NCAM, and validated that this method works. So we treat cells with peracetylated MANNAS, which is the precursor to sialic acid. The cell metabolic machinery converts it to sialic acid and then puts it on the cell surface in all sialic acids, monoSia, diSia, oligo, and poly. We then lyse the cells and we separate polysialylated proteins from non-polysialylated proteins using an immobilized endo-end column that we made. And that allows us to essentially affinity purify polysialylated proteins. The mixture at this point is still quite dirty. Polysialic acid, because it's a long anionic polymer, it tends to bind to all kinds of proteins that would normally bind long anionic polymers, like some of the glycosaminoglycans, like RNA, DNA, that kind of thing. If you try to do proteomics on this mixture, or an immunoprecipitation, for example, you get a mess, like hundreds of proteins. So then what we do is we click on biotin to the azide, and we can purify biotinylated proteins on streptavidin agarose. And this is beneficial because that interaction is quite stable to detergent. And so we can wash away all of the non-specific proteins and then do proteomic analysis. We started by just validating the concentration of peracetylated MADNAS. You can see that by time we get up to about 50 micromolar, we're starting to lose polySia signal, suggesting that we're getting a lot of azides into this and glycan, and maybe the antibody's not binding as well. And then with the streptavidin blot after this procedure, you can see that we are actually getting the modified sugar into the glycan. So we did this with natural killer cell or with NK92 cells, as I said, and we came back instead of with hundreds, just 35 proteins after the proteomics, which was great. We sent it through CRAPOME, the CRAPOME database, which identifies proteins which non-specifically bind to the agarose beads that we use to purify them. And after filtering through CRAPOME, we end up with 9 proteins, the top hit of which is NCAM, which was fantastic. So our method works, and we're still working on validating the rest of the proteins. At the same time, we actually sent MCF7 cells, or we've been analyzing MCF7 cells, and these are really interesting because we don't know what these polysialylated, or we didn't know what these polysialylated proteins were. And again, we came up with just 42 proteins, which after filtering through the CRAPOME database, left us with 7 proteins. And these are what I really want to show you because they're super cool. So these are the 7 proteins. And what's cool is that some of these proteins, like MGAT2, actually showed up in some of the immunoprecipitation experiments that people had done previously to try to identify polysialylated protein. But because it wasn't cell surface, everybody thinks that polysialic acid has to be on the cell surface. That's where it does its role. Because it wasn't, this protein's not on the cell surface, it was discarded as non-specific binding. But it shows up again in our experiment. All of these proteins, actually, with the exception of LRP2, are not found on the cell surface normally. So QSOX2, GOLM4, and AGR2 are proteins that are generally found in the secretory pathway. These 3 proteins we see when we do our ELISA again, come in with that primary antibody that recognizes the protein. We see good signal that is substantially decreased when we treat with endo-N. And so these are 3 totally new proteins that had never been identified as being polysialylated before. And so that got us wondering, okay, so we have proteins in the secretory pathway of this MCF7 cell line. They're not doing anything there. That's not generally where polysialic acid acts. But it turns out that in the MCF7 cell line, polysialic acid is not generally found on the cell surface. It's actually in the Golgi. The green overlays really nicely with a Golgi marker that I haven't shown. But that got us wondering if polysialic acid was secreted, and indeed it is. So if you take these cells and you incubate them for several days in serum-free media, and then monitoring, it has to be serum-free media because fetal bovine serum has polysialic acid in it. So serum-free media for several days, and you analyze the concentration of polysialic acid in the media using our ELISA, you can see that it starts to increase. And we had a look at QSOX2 in the lysate, the cell lysate versus the media, and you actually see QSOX2 in the media as well, which is very exciting. We had a look, we measured the concentration of polySia in a couple of different cell lines to see if this secretion or release of polysialic acid into the media was a general feature of cancer cell lines, and it is. In undiluted, which is the light green here, for MCF7 cells, you can see signal using our ELISA, which disappears when you treat with the endosialidase. And if you concentrate the media tenfold, because it's quite dilute. If you concentrate it tenfold, you can see that there's signal in almost all of these cell lines, with the exception of LNCAP. So it may be a general feature of cancer cell lines, this secretion of polysialylated material, polysialylated proteins. And that got us wondering, can polysialic acid be used as a diagnostic marker for metastatic cancer? And so what we've done here is analyze the concentration of polysialic acid in the serum. This is serum taken at the time of diagnosis of people with breast cancer, different kinds of breast cancer. So this is not a concern. Invasive breast cancer is a concern, but this group of people 5 years later was still alive. So the treatment worked. Hopefully the treatment worked for these people. And then in this group of people, the cancer was fatal within 5 years. So what you can see is there's a definite increase the more severe the cancer gets, the higher the concentration of polysialic acid. This is not a big enough difference to actually be a diagnostic, but it does beg the question of whether cancer cells are secreting proteins that are not normally polysialylated in healthy humans, the way MCF7 cells seem to be. And can those proteins be used as a diagnostic marker for metastatic cancer? And we're working on that right now. So then the last thing I want to talk to you about in the last few minutes is another tool that is going to allow us to try to probe the function of polysialic acid. We know that it's certainly controlling the immune response. We don't know how. We know that it's important for cancer. It's certainly important on the cell surface of cancer, but it's also secreted in cancers. And so we have lots of questions about what is going on, what's happening, why is this happening? But we really don't have a good way of removing polysialic acid from cells. You obviously can do like CRISPR-Cas9 or various genetic techniques to remove one or more of the two polysialyl transferases, but you can't do this for primary cells generally. It's much harder to do this in primary T cells or primary dendritic cells if you want to understand the interface between the cancer and the immune response, or just how the immune cells work together. We do have the endosialidase enzyme, which can cleave polysialic acid, but that protein is expressed in E. coli, it's full of endotoxin, and so it's really hard to actually study cells using that lectin to remove polysialic acid. And so what we've actually done, and this is in collaboration with Steve Wither's lab and Mark Nitz, is to examine the ability of this modified sialic acid, 8-ketosialic acid, to inhibit polysylation in live cells. As I mentioned, polysialic acid is alpha-2 8 linked, and so this should not, any glycan with an 8-keto moiety should not act as a substrate for further elongation. We checked out the toxicity of this molecule. 2, 3, and 4 are just different ways of protecting the sugar, different esters here. And the sugar in this case is they're polysialylated so that they cross the cell membrane. And the 3 different esters are not toxic really in MCF7 cells. There's a little bit of toxicity with molecule 4. They're not toxic at all in this AT-T20 cell line. This one makes polysialylated, lots of polysialylated NCAM. We did a cell surface labeling experiment where we just labeled cell surface ketones using a fluorophore. The unlabeled cells have low fluorescence intensity, which increases in each of the cells treated with the 3 molecules. So it definitely makes it into cell surface sialysides. And then we looked at its ability to inhibit polysylation in live cells. So these are MCF7 cells treated with increasing concentrations of each of the molecules. And you can see obviously in the Western blot that increasing concentrations of the molecule do decrease polysialation. And this is densitometry of these. We could have done an ELISA as well to quantify, but we didn't have the ELISA working when we started these experiments. So we see a substantial decrease, especially with compound 3. 200 micromolar compound 3 for 3 days reduces polysialation to about 20% of the untreated cells. This is with ATD20. As I said, lots of high molecular weight polysialic acid on NCAM. Molecules 2 and 4 don't seem to do too much under the conditions that we tested. But again, molecule 3 seems to be quite potent. At 200 micromolar, we see only 30% residual polysialic acid. We looked at NCAM to make sure that we weren't just reducing expression of the target protein. And so this is NCAM here, treated with the endosialidase. So you can see what the protein actually looks like, and we're not losing expression of the protein. This is NCAM without treatment with that endosialidase. So you can actually see that molecule 3 decreases the molecular weight of that. It certainly seems like it is preventing this protein from being polysialylated. And then these are our controls down here. We did some in vitro assays to determine whether or not the monoSialotransferases, ST3-Gal1 and ST6-Gal1, could transfer 8-ketosialic acid. Our hypothesis at the time was that the ketone was such a small change that these enzymes should transfer that modified sialic acid just as efficiently as the unmodified regular sialic acid. And that actually turned out not to be the case. So if I just draw your attention to the green and the red, the green is activity on a small fluorescent substrate with the regular sialic acid, and then the red is with the 8-keto sialic acid. And it's the same over here. So you can actually see that these enzymes don't like the CMP 8-ketosialic acid very much at all. This enzyme, we couldn't get saturating conditions at all under any conditions, but we did get it for ST3-Gal1, and that's shown down here. And the kinetic parameters say that the 8-keto modified sialic acid is about 10 times less efficient for this enzyme than the regular sialic acid, which is really interesting. We had a look at the crystal structures of the active site, and the ST3 and ST6-Gal enzymes seem to have a smaller binding pocket around that glycerol side chain. And it's possible that the change in hybridization from sp3 to sp2 with the ketone actually alters the geometry such that these enzymes don't actually use this substrate at all. We wanted just to check to make sure that this wasn't being an inhibitor of glycosylation. And so we mixed equimolar amounts of the regular sialic acid and the 8-keto modified sialic acid. And for ST6-Gal1, we still saw the same amount of product. So this enzyme just doesn't see the 8-keto moiety at all. It doesn't affect the ability of the enzyme to transfer the regular sugar. ST3-Gal1 was a little bit different. We actually saw a decrease in the efficiency of this enzyme for transferring, for making any kind of product, even though it still has the same amount of regular sialic acid as it did here. But that's consistent with this being a poorer substrate. And then the last data slide that I want to show you is that this actually works in primary T cells. So we took PBMCs and isolated the CD3 positive T cells and treated them with compound 3 for several days and monitored polysialation. And so if you just look at days 1, 2, and 3 to start, this is the polysialic acid on day 0. We know that this is polysialic acid, or at least alpha-2,8 linked sialic acid, because if you treat with endo-N, you lose signal. And then if you treat with it over the first few days, here's the quantification, you can see a substantial decrease in the concentration of polysialic acid in cells treated with the 8-keto sialic acid compared to the DMSO control. We also wanted to see if this was a temporary situation because we want to reintroduce polysialic acid and provide further evidence that we're not mucking with the polysialic acid pathway or any of these proteins. So at day 3, we actually removed the 8-keto sugar from the media. And what you can see is that the concentration of polysialic acid increases quite dramatically to the level where of the untreated cells. So I've shown you a bunch of new methods that are going to allow us to better probe the function of polysialic acid in the context of the immune system and its relationship to cancer. We're looking to see if we can develop a diagnostic for early detection of metastatic cancer. We're working on identifying the polysialylated proteins in immune cells. And we now have this polysialic acid specific inhibitor that's going to allow us to study the role of polysialic acid better. So with that, I want to thank everyone for your attention. Really like to thank the people who have done the work, in particular, Carmona Hunter and Talia Dirksen in my lab, Nicole Thompson in Warren Makarchuk's lab. Amanda started this project when I was still a postdoc in Mark's lab, and then Xi Zhang and Gao and Hongming Chen from UBC. And thank you very much for funding. And again, thanks for your attention.